the money. Like, okay. Um, I'm back. I have flip-flops. I'm not barefoot. My foot is better. My toe is broken. I wish I had a really good... I still have a good story, like I broke my toe in Brazil. Like, that's a kind of like a cool story, but I wish I broke it, like, I visit a hospital, all that stuff. But that's, I mean, I wish that I had a story like somebody stepped on my foot while I was dancing foro or samba or something. But unfortunately, it's not true. I just tapped into my bed, which is like the stupidest way to break your toe, if you're going to break a toe. Okay, that's not why you're here. You're here because you care about folding. And so I know that you care because you sent me lots of emails. Thank you so much. Obrigada. I'm so happy that you did the homework. I tried very quickly to copy-paste your results to compare them with mine. It doesn't mean that my results are correct. All I did was the same thing as you with one of my PhD students, Nick Judd, so we can just compare notes. But I wanted to, with Carolina, we'll probably think of rewarding people, but I wanted to like single out. So there was Matayos. Can you like wave? Hi. And then I think you're the only one who did the boat 22. So you get something for that. Can you write that down? And then, yeah. And then there's Gustavo. Hey, oh, you guys are sat together. You were copying each other on you're doing cumulative wins. So I think you win because you did the largest puzzle, because you got the 325 right, I think. I'm not sure. We should have, but you made an attempt at something that might be right. And that's a hard one, so that's cool. Then Christina, where are you sitting? Oh, there you are. That you did the most of them, like the number of the ones. You did the smallish ones, but you did the most of them. Huh? In your group. Christina's group. Yeah, we cannot list everyone now, but yeah, the team of Christina. Okay, so that's another round of applause. Then we have Pedro. Where is Pedro? Hi. So you did everything up to 19, but you didn't want to share it with me. Okay, we trust him though, right? He said everything up to fish 19. So that's pretty good. And the fish 19 is correct. And then how do you pronounce this? Jaderson? Jaderson. Hi. So and you wrote down that it's with Luna and Joao. Ah, the three of you. Okay, well done. So round of applause for the 5 teams who submitted their homework. Thank you. I hope you had fun doing it. And I hope you realize that very few rules can get you very far. Keep in mind, for like a chain of 25 or 30 blocks, we're talking about selecting configurations out of the thousands, right? So very few rules, very unique solutions. So let's talk about the results. Atom 7, this was an easy one, right? We agree. There's only one way to wrap around. There was some mistake about, or some ambiguity about what the rule is. So this is actually the HP model. It's developed by Ken Dill as a toy model for physicists for how proteins fold. And the rules are actually, you have a hydrophobic core, so yellow likes yellow. They like to be together. But the blue have to face the solvent. So I think your team, Haderson, I think you made them, I think you misinterpreted that rule. The blues have to be on the outside. They have to face the water. And the HP model stands for hydrophobic, polar. So polar molecules sit on the outside facing the water, and hydrophobic ones sit on the inside. And we'll talk more about principles of folding that are underneath that assumption. Okay, so that's the atom. the diamond structure with 4 of them in the middle. So I'm not sure. I think that the one at the top, if you rotate it, is the same one as the one that I got. And these two are definitely just a rotation away. One question, anyone who feels like answering. Do you think that there are multiple backbones that they can fit into the same shape to give you the same solutions? Or do you think it's one backbone per solution? Did any of you discover? Think about it for a minute. Yeah, actually, I will show you something that contains the answer in it. Look at this guy. The backbone is the sequence, which is given to you here. And you have to go blue, blue, yellow, blue, 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 yellow, blue, blue, blue. And the white line shows you how that person did it. So looking at that, the answer, you should be able to figure out the answer. Is there another way that you could draw the same sequence? No. No, I'm not. I'm saying no symmetries allowed. Can you see one? Yeah? Yeah. Yeah, look. All you have to do is flip this one around. Blue, blue, yellow, blue, 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 etc., etc. So different backbone, same shape, same sequence. So the answer is yes. It's a trivial example, but it shows you that there are multiple solutions in sequence space, in backbone space, that are possible. Okay, that was just a little side note. So the diamond, we had a couple of candidates. I didn't have time to cut and paste all of your results. This one, I don't know who put it, but I don't get this result. We don't have time now to discuss it. Maybe we can have the discussion after class. But you were able to put them all in a row. I needed a kink to satisfy the requirement that the blues encapsulate the yellow. Anyone have a particular one that they thought was really hard? Which one? The Sphinx was really hard, I agree, because it has 2 subsections that require it. Exactly. You have to, no, yeah, they don't all work in the sequence, but there is one that does. So that's the point. How to funnel the conformational space. Bone 17, I don't think anyone did, or at least I didn't have time to copy paste. This one, I wanted to show you, I was very proud to discover that one of them has a chiral equivalent. So if you work with geometry like I do, and if you stare at these things for long enough, you can see that these 2 structures, it's not enough just to rotate. You need to slide one with respect to the other, so they're mirror images of each other. So uniqueness, what did we try to do? We tried to figure out, are these sequences coding for unique shapes? And the answer is yes, but not including chirality. They're allowed to have left and right handedness. Here's the Sphinx. The less symmetric things look, see how it's a bit rugged, weird? The harder it is, because how do you do this? How do you look at this sequence and decide what to do with it? So we're going to talk, I came up with a couple of things you can do to reduce the number of trial and errors that you can do, but I'm interested to know if your brain came up with strategies for how to look for these. You did? You did it in a different way, but you got the same shape. Ah, you got a different shape. Oh, that's great. Can I see?
This is the shape you got.
Yeah, that's my shape.
The sequence, but we got this.
And it fits the sequence. Yay, great. Okay, so we found a mistake in the algorithm because our algorithm spits out the only sequences that give unique solutions. So there must be, yay, this was the goal. The class finds a way that uniqueness is not satisfied. I have to still double check, but that's really good. I'm really happy. By the way, a protein sequence that codes for 2 folds instead of 1 can be very useful in biology because you can have conformational dynamics between them, which if we're not super slow, I will get to in this class. Okay, the fish, you got the fish. And I hope everyone can see the fish swimming here. But it's really cool. So look at the sequence and look at the way it folds. And you already start to recognize that wherever there's a few blues, they go around, they kind of go in a straight line. Then they need to change direction when there's a yellow in between because they need to pack it, right? So when you see alternating things of blue and yellow, you can already start eyeballing the shape you might get. And that's maybe a useful exercise. Cone 21, nobody did, right? Oh, you did the cone 20. Did you get that one? You did. Okay. So Team Henderson gets an extra point there. They get, we'll think of prizes. So we have till the end of the week, right, Carolina? Yeah. Okay. Boat 22, I would say we got the same answer. So you did that, right? So that's marvelous. That's a really hard one. How long did it take you to do it? Really? So it was sheer luck or you had a strategy? Yeah? Okay, that's what you did. You just started from putting them all here and then seeing if you can wrap, and since it wrapped you, or I'm done. You see, that's the thing. When you zoom out and you find a strategy, you don't get lost in trial and error, basically. That's really cool. Anyone else? Any other comments? Okay. Boot. That was a hard one because it has lots of regions. One here, a single one, then the 4. So you pack it in this rugged structure. Okay. This is the tree. You think you got it wrong? It was it, because I thought maybe you labeled it wrong, because I think this might be the 18 one, not the 25 one, huh? Okay, that's one of the rules that's one of the strategies I was thinking. So that you say that you want to minimize the free energy of the interface between blue and yellow. That's nice. So looking for principles of design like that is basically the search for protein folding problem. Okay? That's great. Okay, we're almost there. This is the largest one ever. Even the code doesn't know how to do beyond 31 because we ran out of computational power. Nobody did the blob, but maybe there's a strategy like yours that would give it to you quickly. Okay, did everybody understand why the game is called sequence? So it's called sequence because there are 20 letters in the amino acid alphabet, and there's 26 letters, at least in the Latin alphabet. And so what do you end up with? You end up with some letters that are missing, and you can spell, I made it bold, sequence, but you're missing U. So that's why we changed it to V. I was going to ask you that, but then I realized that by mistake we printed out the answer. So I was like, okay, there's no point in asking. So everybody got that. That was like a historical thing. Okay, so the question becomes, how does this help you become a protein scientist? It helps you because it makes you understand that what's actually happening in protein folding. You have a chain of a polymer. You have species that like each other and want to compact, the yellow guys. You have another species that are the amino acids that like to be exposed to water. You have something like a hydrophobic collapse. But what's really happening that's interesting in the physics point of view is that you're obviously minimizing energy and maximizing entropy, but how? You're doing it by thinking about the solvent. So the exclusion of this, you have this chain, it's exposed to the solvent that has a lot of penalties because you have hydrophobic regions exposed to water. And then there is a sort of phase transition. And the phase transition takes place because these oily bits come together in a blob. It's called the molten globule. And the hydrophobic interactions are intact. And then you essentially, at another energy scale, start to sort them out to get the right fold. This is talking about biology, how we can start to understand it. I'm only using words now, but we're going to go a little bit more in depth as to what kind of tricks you use. So I didn't make slides about this, but maybe it's worth mentioning. How can you even think about folding? What's the main historical result? I don't know how many of you know about calorimetry, but calorimetry is basically a way to measure specific heat. So for instance, if you take an ice cube and you heat it up and you want to observe the melting phase transition and you look at how fast does the temperature rise, you put some power into it, some energy per unit time, you watch how the temperature rises, and from the gradient of that temperature rise, you know the specific heat of your material. And that's a very boring experiment, and you're heating, the temperature is going up, but then something amazing happens. The temperature stays the same, you keep heating, nothing happens, and then you're melting the bonds. That's called the latent heat that you need to put in to melt the ice cube. And it's to do with the fact that you're going through a phase transition. And so if you do that with heteropolymers or diblock copolymers or anything that isn't an evolution designed, let's say, protein sequence, nothing exciting happens. The gradient of the temperature keeps going up, and that's it. There's a specific heat and so on. You do it to a sequence that's a protein sequence. This is a few Nobel Prizes. I'll explain which ones. But one Nobel Prize is to say if you do calorimetry, you find this amazing result that proteins have a latent heat and that you keep heating the protein. The temperature doesn't rise until you melt the folded bonds, and then it's an all or none transition, and it's modeled as a two-state model. And this course is too brief, you know, to actually go into all of that and how you get the two states, the unfolded and the folded state, like you did for homework, and what is the energy. But basically, the energy difference between the unfolded and the folded state is given to you by the measurement of the latent heat, okay, in heating it up. So then another mystery is, so there's three names. There's Amfinsen's dogma by Amfinsen. There is the Leventhal paradox, which is about the vastness of this configurational space. And then there's all these experimentalists who actually showed that even if you take a sequence outside of the cell with no biological active energy input, these protein sequences, polypeptide sequences, spontaneously fold into their correct architecture, the same one as they have in the cell. So it's kind of amazing that no cellular machinery is needed for the folding of long chains. And conformationally speaking, that search is incredibly difficult and almost impossible. But there is some principles of this giving energy to the solvent, minimizing the energy, maximizing entropy, making the globule, that drives the system, exactly like if you watch a crystallization process or something like that. So lots of physics behind folding. It hasn't been solved from the physics perspective, but let's talk for a minute longer. What did you learn via this hydrophobic polar model? Are there different backbones? We said yes. Are your foldamer shapes unique? You said no. That's great. I was really not expecting that. How many enantiomers? I think we only found one pair. What are the strategies you came up with? So you said, how would you take it as hexagons and try to minimize the exposed structure? So what I said, and I'm going to, okay, anybody else found a strategy? What worked for me was to forget the sequence, take it as a gas, look at how many shapes I can make with all yellow together and all blue around, look at which ones of those look the most compact, and then look at the sequence and say, oh, no, it's not a block of yellow and a block of blue. There's ruggedness in the sequence, so I gotta go find a shape that has some kinks in it. So I sort of went backwards, and I wrote that, I don't know if it's clear, make a full set of possible shapes that fit the rule 2, right, that's that. Find the most congested section of the chain where the color variance, the changes between yellow, is the highest, and look at where the subshape resulting from the folding could fit in the set of those possible subshapes, if that makes sense. So relating blobs to sequence lengths. That's how I tried to do it, and sometimes it works, sometimes not. Any other ideas that popped into? It would be nice if there was a rule that we could apply and do it something really elegant. Okay. And I also found that warnings are you can make shapes that have blue tiles completely surrounded or not touching yellow. And that often made mistakes. Okay. All right. So let's review for a moment, how can you think about this in terms of the phase space? It's sort of a silly idea, but it's called a paradox because it shows that if all the configurations were equally probable, if you're on a flat landscape, no favoritism, then one way you can think about folding is just counting. What are the number of possible configurations for a chain of length n? And what is the time for configurational transitions? How quickly do atoms or molecules jiggle, right? So you can do a little calculation and you say, I have 3 degrees of freedom, which are the dihedral angles that the protein can sit in. I still have my block chain. And then I have, where is my snake? Well, anyway, and then I have 3 to the n possibilities, which in this case, if you're interested, you can look at a beautiful paper, very short, about loop fold structure of proteins, the resolution of the paradox. You take a chain length of a small protein, 150 residues, you take 3 to the n, you multiply it by the attempt frequency of the jiggling of the molecule. That's 10 to the 12 attempts per second, and that's generous. It's probably much smaller than that because proteins are bulky. And what you end up with is a number that's longer than the age of the universe. And so the idea becomes, how do I, even computationally, I cannot enumerate the space, so I can't solve the protein folding problem analytically or numerically or using theory, so what can I do? And there the idea becomes that you should think of this as a multi-dimensional complex energy landscape within which you don't know what the reaction coordinate is. And that's the real big problem with proteins. You don't know if it's the end-to-end length that matters, or if it's the surface exposed to the solvent, or if it's the interaction between different specific residues that need to come together. You don't know what rule nature has for which reaction coordinate is the one that's optimized. And so people have struggled with this for, and this is from the 1950s. There's one Nobel Prize to Amundsen for the story of specific heat and the fact that it looks like a phase transition. There's another Nobel Prize given to David Baker and his team for saying, screw the physics of this, let's do AI. And this is AlphaFold. And so I want to just show you that even though the curse of dimensionality kills you when it comes to the Leventhal paradox, you can do some things with toy models in physics. And what do you learn in physics? You learn that you need a landscape that satisfies three things. One is uniqueness, so that you actually can control the geometry. One is smoothness, which is basically another way to say that the landscape has to be funneled. It's like crystallization. Once you form a nucleus, you keep growing and things are attracted in the right way. And so you need some design that makes you avoid kinetic traps. So we can call this like you need a smooth, downhill landscape towards some unique solution. And you need a third thing for biology, and that's called robustness. And why do you need robustness? You need it because if you want to avoid disease and if you want to prevent function, you need to have a system in which this sequence is robust against perturbations, so that if I change a few mutations, I don't get mad cow disease or Parkinson's or Alzheimer, etc., etc. So those three things you can demonstrate with toy models in physics. I want to show you the AI result of how you can use AlphaFold to predict protein structure. But there's a real gap in knowledge between the toy models and sequence to structure prediction in the way of principles of design. You see? So I'm not saying that our game or our research even will contribute to that, but at least that's the hope. Okay. So here is one slide I made for the AlphaFold. There's now versions 1, 2, 3, they keep improving on it. But the idea is quite simple. What they do is they say we have about 50,000 proteins that we know the structure of. We know their sequences, and we know who likes who and how much. So these are a bit like the matrices I showed you for our toy models. And they can simply do sequence alignment, and they can say, if I see this amino acid in proximity of that one, what's the likelihood that they'll be at that distance with each other in this 3D geometry? And then they compare what the force field MD data shows them, what the protein data bank shows them from the X-ray crystallography structures, and they can then change the sequence and ask what would this correlation matrix predict for proteins that don't even exist. And so it's really cool, amazing work whereby they make synthetic engineered proteins that don't exist in nature, and yet they fold exactly according to the rules that the AlphaFold predicts. And of course, they use machine learning then to iterate on pairwise distances. So they have 3 different correlations that they try to implement together. And I was hoping Mathieu would be here so that if there are any questions, he can answer, because that's kind of the limit of my knowledge. But it's data-rich inference. Yeah. All the known protein data bank, all the experimental structure to function, sequence to structure correlations. They plug it in, and then they even have ways, this is why it's AlphaFold 2, I was reading what's 1, 2, 3. They have ways to constrain further and say, oh, you predicted something that's actually not compatible with the charge, the way the charges are distributed, or some physical constraints on the model as well, not only sequence alignment. Okay, so going back to our experiments, going back to our lifelike colloidal story, just to refresh your memory, we found these 12 foldamers that we were able to reproduce experimentally, and we really liked the idea that the algorithms predicted something we could make in the lab by designing DNA sequences and binding droplets together. Interestingly, n equal 12 is the only chain length for which there was no solution at all. That's why I put a cross there. And I bring this back up because I could have given you a different game. And that game could have been to say, look at our Nature paper, look at this lexicon of folded structures. What is the protocol that we should have used in order to get there? And you could have been able to figure that out using some pretty basic rules. And again, it mimics a bit the collapse, some kinetic arrests. You can talk about entropy by counting the number of backbones that can fit into each shape. But I went back to this because I wanted to highlight something a bit poetic and a bit philosophical, which is, to my mind, even more important. It's like all of this research is based on inventing games so that you can ask some precise questions. And one thing that we learned is that we be patient toward all that is unsolved and try to love the questions themselves. And so we've been enjoying the questions, and we've treated them like locked rooms and like books that are now written in a very foreign tongue. Live the questions now. Then gradually, without noticing it, live along some distant day into the answer. I really like that. And it's really something that retrospectively happened in our 20 years of trying to, because we start with mayonnaise, we have oil droplets in water, we have a bucket of them, and we're like, what do we do with it? How do you make science out of that? How do you we'll program life. What does that mean? I'll read some books, But after a while, it's like I'm not pressed by what I want to answer, but I'm pressed to care about the questions themselves. And so I wanted to just take a moment to stress that you shouldn't stress about the answer. You should stress about the question. Okay, we move back to actual science. So one of the things I lied to you about quite explicitly is that these unique solutions in the downhill case can get you to the structure that you want. And I did that for simplicity because I knew that I was going to correct myself. So the yield of these structures can be very, very poor. And the reason is that even though it's the ground state, even though it's unique, it's kinetically not accessible. This guy, the funnel smoothness, just doesn't work for you. And so we spent the last couple of years studying how we can do a non-equilibrium drive modeling, in theory, to try to predict what's the best drive through the energy landscape to get to a given structure. And so I want to show you that quantitatively, and I want to show you, again, very basic examples of that. So I already showed you how a 7-chain can fold into a rocket ship. If the blue-blue comes together, you form 1, 2, 3, 4, 5 bonds to make it. Of course, by now you already know that there's lots of other pathways. And more importantly, you have these kinetic traps from which you cannot be rescued. So one of the things we've been interested in, can you build catalysts and chaperones to rescue traps? That's one idea. But we've also dealt with trying to say, so how could I do it? I could do it 2 ways. I could be like, oh, you're going down here. I'm going to tilt the temperature in response to you going down the wrong way, such that you don't end up here, but you go rather this way. But that would require a relationship, a communication of which pathway you're taking and me modulating it. And that's usually the way the drives in the literature work. But what if you wanted an average answer? What if you wanted, like, given all these pathways and the flux, the probability flux through the network, what's the best way to optimize a given structure if all possibilities are possible? And very quickly, the math gets horrible because you have downhill rates, uphill rates. They're asymmetric because downhill you're diffusing, and if you meet and greet your partner, you bind. But then once the bond is made, you have Arrhenius to go back. So you have diffusion downhill, you have Arrhenius backwards. And so I want to talk to you about Markov chain models, which is a way that you can model this tree mathematically so that you can predict which temperature protocol would optimize the yield of a given architecture. It's a non-trivial problem to solve. So if we look at a tree that's really simple, just 5 bonds, this is a homopolymer. Everybody likes it. I'm gonna show you something very boring, and then I'm gonna use that to answer something a bit more fun. So in this case, everything is boring. There is one folded state, the pentagon, another rude word, and then the chain up here, and just a few states in between. And then you can model this process by saying, I have downhill rates that are diffusion limited, so my rates are basically proportional to the temperature, and I have uphill rates with dashed lines that are Arrhenius-like. And I have a probability distribution for every single one of these states that evolves with time. So I can plot the histogram. What's the likelihood of you being here at time t, et cetera, et cetera, et cetera, for each one of these states. At the beginning, it's going to be everybody up here. At the end, it's going to be everybody down there. And I write down the Markov chain model in which the time evolution is essentially given to you by this transition rate matrix. So if I know the downhill rates, if I know the uphill rates, if I know the temperature at which I'm working, I also know the probability flux through my tree, right? And now the question is just how many rates are we talking about, and how big is the tree, and how difficult is it to solve? And so we optimize the parameters, the rates, and the temperature protocol to fit the data. Now, since we make an assumption about the temperature dependence, we only need to estimate one set of rates which should obey the temperature dependence for all the data. And now we've got options. We can either go to the lab and we can collect data as a function of temperature by folding these guys, or we can do MD simulations, and in this case, I'm going to show you that both of them work. So the data is probability distribution of time for every single state. Here you see the disappearance of the chain with time, the appearance of the pentamer. And why isn't it 1? Because we're at the wrong temperature. So here is the model. So what do we want to do? We want to minimize the loss function such that the log of the probability that I'm actually measuring over the log of the probability that I'm assuming is essentially being the same. And so the data is in the data is the dots, the model is, the fit is the line. You see a very nice agreement, and now you can ask, do the MD simulations and the experiment follow the right temperature dependence? So we vary the temperature, we don't vary the rates in the optimization.
Yes, you say that...
and everything works.
Wait, I was lost.
Good.
Points are
Data, yeah.
experiments, and
That's
then you
right.
have this the line.
The line is the Markov chain model, asking,
Okay.
is it okay to model who knows what goes on, right? You put a chain of droplets there and you watch it fold. Now you make these assumptions about Arrhenius diffusion, about even that the tree is correct, and that the right transitions are allowed, not allowed.
I see, but this is what you are measuring is the probability of having this structure As a
Yes.
function of time, that's what you say.
Correct.
Okay.
And so this one goes from 1 to 0. You know, it depends on the temperature. So you see here that here you have the temperature dependence that is captured by the model. So that's kind of nice, but it doesn't teach you anything. Just says the model does a good job of fitting the data. Now, how can I make this useful? How can I predict something I don't know? So if you just make this chain an alternating one, you already have only one shape. These are the pentamers, but now you have 4 different color arrangements, which are all distinct. And if I ask you, how am I going to, I want a specific color arrangement, because I'm a protein and I need to fold in a specific way, then a couple of solutions are, again, trivial. If I switch on yellow, yellow, then blue likes yellow, then blue likes blue, I get only this guy. 100% yield, nothing. And what's even more fun about this one is there's really only one path available. So that's it. Second one has 2 paths, but they both end up in Rome. and then, so I have no further work to do. If I go to this guy, I hit this kinetic trap, which I call the bow tie. And the bow tie screws me up because I can't get 100% yield of this guy. Moreover, I end up here. It's a dead end. And so now I can ask another question. I can say, I can optimize the temperature protocol and these epsilons of what's the energy of attraction between blue-blue, between yellow-yellow, between blue-yellow. And so I add more parameters, but I do another loss function, and I say, can I optimize this guy here for which temperature protocol and which combination of energies of interaction do I get the largest yield of this structure? Okay? And now since I have the rates, I actually can solve that system. So let me show you the results. The results are, here is the temperature protocol. If I make the blue-blue bond completely irreversible. So 30 kT means once it's done, it will never undo. And if you watch the video, you see the flux going down here. And you see that I get stuck in the bow tie. And the bow tie is chiral, so there's a pair of bow ties. If I plot the probability as a function of time, I get stuck at 62%. Now I do something fun. I say, I can optimize the blue-blue bond to be reversible. Now watch this. The flux from these guys is going to become a dashed line, which means it goes uphill. So I'm rescuing slowly the guys that stay here. And since they're higher in energy, they all eventually end up where I want them to go. So just by cycling temperature, or just by picking 6.3 kT as the interaction energy and working at the temperature that the protocol tells me, I get 100% yield. The bad news is I need to wait a really long time for that to happen. Yeah. No, I wasn't specific. So there's no temperature cycling. It's actually this protocol here that switches on one temperature, then a second, and then a third. I give it a step function. You can input any function you like. You could even make it cyclic, but in this specific case, it's a staircase of temperature. And then I make this window of time longer and longer. That's this x-axis. And I ask what's the optimal parameter for the epsilon. And I get 6.3. Yeah? Yes? Yeah, it's always from T0 till T infinity in the experiment. The blue-blue bond is always the same epsilon. So that's what the point I made at the beginning, that you optimize averaging over the entire tree for all pathways for all time. What you could do is like interrogate the system and keep changing and say, no, you went down the wrong way, heat it up, you know, and stuff like that. But we don't want that. We want to have a protocol for which it's optimized for everyone. Yeah?
Sorry, okay. I see now from the going of the probability with time that there is, let's say, a transient initial time, let's say, which is not properly described by a Markovian model, which I assume is because of the granularity of the state structure, let's say, no? For the fact that you need some time to leave the initial configuration, have a partial reconfiguration, and then reach the other configuration.
Yeah. Every single transition is associated with a rate.
Okay, but in the picture there is no partial reconfiguration picture, right?
It's per bond.
Okay.
Every single bond creation event has a state associated with it, and so on.
Okay, okay.
But nothing more coarse-grained than that.
Okay, okay, thank you.
You can't make simplified trees with more coarse-grained things for larger. I'm showing you the simplest example there is. Any other? Yeah?
Maybe I missed it, but I mean, I know you're not changing the protocol, but does the protocol shorten? It is a question if it could shorten the time we need,
To wait.
To wait, yes, to have 100% yield.
Yes, this is going to be my next slide. So that's how can we? So we've optimized the yield. Thank you for that. It's always nice. But we haven't optimized efficiency. Is there a way that we can make this optimization work for us so that we optimize the product, but we also optimize folding time? And here's a counterintuitive thing that we discovered really just by playing. And then we said, oh my God, that's what happens in biology, kind of. I mean, everything we learn here is only kind of true, right? It's just very simplified toy models. So, if you look at the tree, to get to this structure, the one we want, the triangle in the middle, you don't want the yellow-yellow bond at all. It's actually wrong. It's taking us down pathways that we don't want. But if you allow weak interactions that are the wrong ones, they rescue folding. They don't make you go down the wrong blue ones. You see what I'm saying? So for instance here, we say make the blue becomes irreversible, but the yellow is now doing the rescue operation. So a very weak wrong bond actually gets you still 100% yield, but shifts your folding time by an order of magnitude. It's such a small system that, you know, you shouldn't read too much into the actual values, but the principle is nice. It's to say, how does that work? If I play you the movie, you will see what happens. Did I already play the movie? Yeah, I did. Hold on. Look, the yellow, you even get a wrong ground state, but since the yellow interaction is so weak, the flux is immediately bringing you back to the right one. And so the wrong weak interaction makes you avoid the bow tie, and you end up with much faster folding. And this is exactly what happens in biology. There's a hierarchy of energies present in the system: hydrogen bonding, hydrophobic interactions, specific disulfide bonds that are almost covalent in their strength, and so on. And that really helps you with this funnel idea. And so actually just making things blob into a molten globule that's wrong, non-specific, it actually makes it faster for you to find the right partners to get to the fold. So it's kind of a neat result with a toy model. The problem with Markov chain models is that very quickly you run into, again, this curse of dimensionality, which is that even an n equals 6 already has a much larger tree. So we've been playing, and I didn't make slides on this, but we've been playing with the idea, is there a way, either using machine learning or physical intuition, to classify different transitions into classes so that we don't need a rate for every line, but maybe for a set of lines. So we can reduce the parameter space so that we can solve larger systems. And I don't have results on that, but it's just, how do we scale up? That's a bigger question. We don't know. And again, you know, that's where the whole, how do we build bigger things, bigger functional things? So this is nice. This is just to show you that the green guys are the optimized ones. We can always get a better yield. We've done it for n equals 6 as well. Maitane and Zorana's group, to close the loop, we said, oh, okay, we have a Markov chain model, we have data, we have a prediction of what to do, we run, and then she takes that result, she runs the MD simulations back to say, do we see an improvement? And we see the model is in good agreement. The blue and the green are nicely aligned, and in the MD simulation, we see this working. Okay, what do we do with, yeah?
Puzzled about the temperature issue, because, I mean,
Sure.
I mean, if I think about Monte Carlo, if I turn temperature to 0, then I will get stuck, and then we can
That's the
raise the
downhill.
temperature, and then you are going to sample more your conformational space. That's what you are kind of doing when you allow the system to come back, right?
That's right.
But now I'm thinking that you were comparing with experiments, but in experiment, you cannot go to temperature equals to 0. So you are always allowing for, no?
That's wrong, in a sense that I can just go to a temperature in which the
Okay, small enough.
where the melting temperature, where I work below the melting temperature enough that the bond is always on.
So you actually manage
So that's
to,
really downhill.
okay, you can do this.
So downhill is the easiest for me because because the melting temperature of DNA is very sharp. It's like a degree Celsius. So if I'm 5 degrees below, my Boltzmann factor is like, it's exponential. So I never see it go back.
I see.
I know
Okay.
it's completely irreversible. What's harder to tune is the amount of reversibility.
Okay.
Like the epsilon of 3 and the epsilon of 6, that's what they predict in the model, in the optimization. How am I going to do that in the experiment?
Okay.
That's hard. But we'll try. Well, I don't have results. This is all unpublished, by the way, just for your reference. Okay, because it's the stuff I'm thinking about right now, so then it's, you know, it's good to talk. We learn from each other. So, why do we want to do this? We want to do this because our idea is to start with these little triangles, pentagons, whatever it is, and then glue them together to make mosaics or to make these dimers or self-limiting structures. The problem is we really would like sequence control. So for the few of you who are experimentalists, I wanted to show you something cool from the experiment, which is that you can control the amount of DNA to control how many partners the droplets can make. And you can start from a gas and you put the magnet on, and what happens is that there are invisible magnets in the water that are nanoparticles, so 1,000 times smaller than the droplets. And because of the magnetization of the ferrofluid, if you like, the droplets, which are non-magnetic, actually align. And we can do this in a way that randomizes the sequence because we can change the temperature and glue and work above the melting temperature here, so that they look like they're bound, but they're not. But we cool it down, the DNA hybridizes, and then you have random sequences. So it's a nice experiment in which what you see here is freely jointed chains with different sequences, and then now we're playing with the binomial distribution. We're really going to basics. Binomial distribution, change the composition of blue and yellow to get the different sequences. And this is, I wanted to pause for thought. I don't have an answer to this, but we had one thought, and I wondered what you think about it. So what we find in simulations, in Zorana's simulations, is that if you look at chain length, let's just look at the longer ones. This is a bit tricky, this graph. I call it the grandpa jumper graph because it's made of colors that maybe your grandpa would wear. But the blue here is always the largest, meaning how many sequences fold for a given shape. And what you find is that there's favorites. Many, many sequences fold for the same geometry. And that's weird. We didn't expect that. So it's not an equiprobable thing. It's not, even when everyone likes everyone, and you just say, okay, I want to do sequence with my protocols, what kind of shapes am I going to get? And what you find, in fact, is that, for example, n equal 12, most sequences, like 80 of them, fold only this one blue guy. And then what you see here is orange, red, and so on, is one other geometry. Just one geometry is red, one is blue, one is... So either you code for... It seems that shape is somehow very robust. And so the question in the biology literature is, why are there so few protein shapes? There's beta... there's alpha helices, beta sandwiches, turbines, there's beta barrels like GFP, but there's very few shapes. And the number of available configurations is like, age of the universe, blah, blah, blah. So how do they get selected? And so there you come with this, well, there's evolutionary design, but why to design function? But the idea from the toy model would be a simple one. It would be to say, it seems that most sequences anyway favor the folding of given shapes, which means that if you do random selection, every generation further, you will have more and more of the favorites and less of the ones on the edge of the distribution. So it gives an argument for random selection with no bias of any kind towards function. In sequence space, we'll have favorites in geometry space. So we thought that was kind of a fun. Did that make sense, Carolina, or did I not say it? So think of this as number of sequences. It's number of sequences. This was a misnomer. I should have changed it. It's kind of the same. This is the chain length. So don't even think about this trend. Just think about the fact that there is always a favorite. There is always the most number of sequences that code for the same geometry. That's all that this is teaching you.
Yeah, but for example, is it the 12 case?
Yeah.
What corresponds to the blue one?
So I should have drawn it. It's like a structure that's one shape, and all the sequences code for that guy, for that shape. Different color arrangements, given a set of rules. And then very few sequences do something different. And for every n, it seems that there is favorite geometries. And if that comes, say, randomly, if that arises naturally because of geometric symmetry and so on, constraints like the type of arguments we were discussing, hexagons, this and the other, then maybe there's just a natural reason for why, a physical reason why it's happening. Yeah.
I guess maybe I haven't thought too hard about this, but couldn't there be some sort of an entropic argument for that? I mean, for a given...
In sequence space, right?
Yeah, I guess that would be the... Well, actually, no, I was thinking in the other space, and if you know some geometry has some, I guess, quote-unquote uniformity, there's probably a lot of... some regularity, some symmetries, there's probably a lot of...
Like, for example, if you take n equal 8 and you take the hourglass, the hourglass is completely symmetric. Then that one, because of its low entropy, because it's so symmetric, is higher in free energy. So in principle, it's less favorable to have a structure that's highly symmetric than it is to have a structure that's the rugged, whatever we had, the Sphinx or whatever, right? Because in the Sphinx, you have something that's not symmetric, so there's many more ways. So these are the type, I don't have answers to any of these questions, but it's, again, loving the questions themselves. How can we formulate this into a physics question of like counting states and learning something about, am I trying to have if I have the most symmetry, then I have the least entropy, which means I'm higher in free energy, but at the same time, I'm confining myself to fewer backbones, so maybe it's a way to funnel. So these are the types of thinking that we've had. Yeah, sorry.
Thank you. Is this mechanisms that leads to robustness in your system?
This is definitely that we would argue is, how do you call that, compatible with robustness, because it says that you need much more than one mutation to change shape. Because all these sequences, oh, I got a mutation, but I don't care because I went to the same shape. Afterwards, you can argue that the color arrangement matters too, and then it's a different argument. That's a good question, actually. Let me show you 13. 13 is, actually, let me not go. If you remember, 13 was the one where you begin to have holes. So you start to have very different looking things when you go to 13 and above, because you make wholly rigid structures that are fully folded, nowhere to go, solid, but they have holes in them and so on. So it's another complexity. Yeah, I'm sticking to like a small narrow where I can define the question, because as soon as I complexify, there's all these competing things that blur the question. Yeah.
Number of chains.
Mm-hmm.
Chains. Like for example, the 12 and 13. The blue ones, they are similar in some way or not?
Oh, you're saying, can I scale up? That's a really good question. So,
Sorry.
the question is, can you learn something about the fold for n equals 6 or n equal 8, and then guess what n equal 14 will look like? Is there some self-similar or other similarity in the way you can build these blocks? And actually, the answer is qualitatively yes. We see, for example, I've been looking at these for a long time now. I can actually tell you what the winner will look like, although I don't know why. I'm like a machine learning algorithm, like it's black. Because I think, oh, all I need to do is add another triangle with a blue and yellow, and that's going to be my winner. But, I don't have like a good answer to it. But they definitely, lots of the winning strategies can fit into each other and enlarge. But not like a rule of growth, or like, that would be a nature paper if it was like something. And we haven't discovered something like that yet. All right. Okay, moving swiftly on. It's very related, don't worry. I'm not going to go ADD on you. So it's about the idea, how can you go from 2D to 3D? So we've tricked the system already because we've made things that are 1D to 2D. But now we want to do 2D to 3D, for example, to make a capsid. So I told you this in the last lecture. Other people have used DNA origami, or Sharon Glatz's group, Patchy Particles, or Francesco Shortino, or Peter Schultz. Lots of people have tried to do building block strategies to make things that make shells. And so those things have their own pros and cons, but if you want to reduce the complexity, we came up with the idea of combining two chains that say make a triangle. They make an icosahedral net with the same rules that you used for folding. So you say, I don't know, blue likes blue, yellow likes blue, you fold the triangle, and you keep those DNA interactions on and you glue the triangles together. Now you can ask the question, if I glue two triangles together, do they make an icosahedral net? What does that mean? It's a little carpet of droplets that can make a perfect icosahedron, just hypothetically. And how do I trick it to do that? Well, by depletion, I put some cargo that likes everybody, and then the little carpet folds around it. And so we've been playing with that for a long time now, and there's lots of detail that I'm going to skip. But our idea of like lifelike materials is you go from sequence to the capsid, and then we're going to try to open and close it to have it be its function. And then you can do adaptation by modifying the sequence to make that faster, slower, whatever you need. Okay, so everything you learned so far, I can go fast, I think here, you just apply now to secondary interactions. Instead of making foldamers, now we're making supracolloids. And we take two triangles. If yellow likes yellow, these are all the nets we make. And here I chose you one example in which every single net along the folding tree, the idea is the same. Every line, every row is an extra bond. There is a ground state, etc., etc. It's all the same story. But now these are all foldable into icosahedra. You're going to ask me again, how many of them are there? There's only 26 icosahedral nets. And there's many more nets than that. So this is a special example where everything is icosahedral. And for example, if you put two triangles together, everybody's icosahedral, but the best case still only gives me 90% yield. Why? Because when I want to fold the carpet, they don't find each other. Kinetically, sterically, it's very hard for them to find each other on the droplet. Whereas, if you take the chevron, that's this slightly different, I fold these guys instead of the triangle, I have many more nets that are not icosahedral, so they can never form a capsid, but I can get 100% yield by optimizing interaction energies. So everything I showed you so far, without going into a lot of the detail, I can now apply to the next level of the hierarchy, and I can make a geometric constraint, which is the one of the wrapping in the red, to make a targeted structure. So we haven't been able to do this in the lab yet. We just have very specific recipes for how to do it using simulations. Okay, we get to active matter. We get to some non-equilibrium ways of perturbing. How can we open and close this capsid? How can we make dynamics? Everything we've done so far, when it gets to a rigid structure, it stops there and it's dead in the water. But there's really cool research on making molecular circuits, and this is one molecular circuit. So I want to spend a moment explaining to you what this pen toolbox is. So we have oil droplets and water in the bucket, but now we're adding three enzymes. We're adding polymerase, we're adding exonuclease and nickase. And those three enzymes have very different interactions with DNA. One cuts it, one nicks it, and one builds it, one grows it. And so what you can do, this is a technology invented by, Guillaume and Yannick Rondelez at the ESPCI. So while I was there, it was a good sabbatical. We started this collaboration where we combine these enzymes with our emulsions, and I'll show you some really cool things that we've been able to build. It's a little bit off the topic of folding because we haven't done the folding with this stuff, but it's all very much along the lines of what we've been talking about. So Before I go into detail of how this works, let me just tell you that there's very interesting work on how you can use molecular circuits and logic gates to control matter. So for instance, if you make predator-prey circuits, where if you create one type of DNA, another type of DNA eats it, and there is a competition between them, you can get oscillatory dynamics that you can program into your system. So now you can think about making circadian rhythm for your colloids or for your material, because you can, oh, by the way, I forgot to mention, in the soup you have bases, and you have ATP, and so you have energy that allows you to keep this going. You can also detect COVID, for example, here, because what this is basically a little PCR machine. It amplifies the DNA. So if you have a little bit of a cold and you sneeze on it, it will amplify, and you can detect small amounts of DNA. And that's really what we want. We need to replicate so that we can make lots of the stuff, so that we can have macroscopic amounts. And this is the project of Lucas Sidenier, who's now in my, he came from Paris. He got a postdoc in my group. And what I'm going to show you is exactly the game that you did for homework, but now we're going to do it in situ using the droplets by changing the interaction matrix using the enzymes. Okay, so it's a much higher level of experimentation. So I promised you to explain how it works, so I'm going to do it here. It's easier to have less information. So you start with something that we call a template strand. So it's got a double-stranded piece and a single-stranded piece. And this pen here is the polymerase. What does the polymerase do? It scoops up the bases from the soup, and it writes the complementary strand to this guy here. Tick, 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 tick. And that's what's being written. Then the nickase comes in. There is a nick, a kink, in between the red piece and the yellow-blue piece. It recognizes that and says that's where I should cut. So it nicks it here, which makes that guy go into solution. Now, when this guy goes into solution, you've got two things you can do. You can add the exonuclease, which chops this guy back up, so you go back to raw materials. Or you can not have the exonuclease, but you can do competing strands. You can, you can have exponential growth of the strand that you want. Why? Because this is actually a little PCR machine. Every time you start with one of these, you produce one of those. So it's a 1-to-1 ratio. But look what happens here. If I now make, for every red guy, I make an autocatalytic cycle, or I grow another red, red for a red, red for a red. Now I have from 1 I go to 2, from 2 I go to 4, from 4 to 8. So I have exponential growth. So by having two circuits, one for the autocatalysis of the template, and one where I have the amplification of the strand I want, what I end up with is a really cool mechanism for an on and off switch for whether my droplets bind or not. I hope I'm not going too fast, but it's how many of you are comfortable with PCR? Not so many? Okay, maybe it is a bit too fast. But if you don't need to understand it fully, just think of it as a way to program in the interactions over time.
What is the off switch here?
The off switch is the exonuclease, which comes in to degrade the strand that you've made.
Thank you.
So you can tune, and I'm simplifying a lot. You can tune lots of things. There he is. Okay. Now, the blue and the yellow bind when this guy is made by this bunch of enzymes. So let's look at how that works. This is now in the experiment. This is a confocal microscopy movie. You see the droplets come together, and you see the recruitment of DNA, and you see how it becomes brighter. We follow the intensity of the patch as a function of time. And what you also see is that it goes up and then it goes down. So you can follow the production of the strand, the sticking of the droplets, becomes more adhesive, and then they get degraded, it becomes less adhesive, so we can actually have real-time information. Now, I don't have time to explain this. I'm not going to go into detail, but we have a paper just published now, I think in February, showing that if you know the size of the adhesion between two droplets, and if you know the number of DNAs from the intensity, which you can calibrate, then in this equation, you know A, you know N, number of DNAs, area of adhesion, area of the adhesion patch, that's why it's called AP. You can then infer what is epsilon, what is the effective energy of binding per molecule of the DNA. And so remember how I was telling you I want to tune epsilon for my things? Now we use this from the experiment to have an estimate for the free energy of binding. So it's pretty cool. I don't know if it will work, but at least we have a way forward. Okay, let me show you we're getting like saturated with ideas and things. So the nice thing is that the remainder of the time, you can treat this as like popcorn movie watching. I'm just going to show you what we can do, and you can enjoy the program. So, and then if you have real questions, we can discuss at the end. So, for instance, here, you make the blue-yellow strand and you heat it up, so that's an on and off switch. So here it goes. They come together, the blue likes yellow, they make a checkerboard, and then they come apart. You get good at this, so you control how many of each of the bases you put into the solution in micromolar units, and you can see that the clock can be prolonged. How long they stick together, how long. Now that's cool for a physicist, because you can also think of this as osmotic pressure. So you can think about it as, I switch this on, my guys come together because they like each other, but now if I had this in a semi-permeable membrane or something, I could get that the water comes in to dilute them, and maybe I can cause the explosion or something, you know. So we're trying to play around with the idea of what could this be function for in the future. Here is the first, you can tell all your friends, I saw the first circadian colloid today. Where is it? And we only have one instance where it really worked, because it's very difficult to control. But look at this. They come together, they come apart. They come together, they come apart. Why? Because in the black, in the water behind them, there's all these enzymes that are working and doing their thing and so on. And what's even more remarkable is that's not the program we put in. So we got oscillations because there's some other stuff in the dynamics of recruitment, adhesion, you know, it's a complex system. So we're trying to understand why do we get the clock and how can we control the frequency of the beats. Anyway, it's really cool. Okay, this is too complicated for now. I know how to simplify. Okay, here, if you make a little you can play with the design of the DNA. If you make a kinky DNA where there's a little piece of it that's mismatched, then you're kind of making the reaction less favorable. So you can just by tuning the amount of mismatch, 10 nanomolar to 20 nanomolar, you can make, for example, the order of which DNA is created first change. So what you see here, if there's nothing, the blue-yellow and the yellow-yellow come together, you make a checkerboard. If you then increase, you end up only making the yellow and you kill the blue completely. here, the 10 nanomolar, you first make the yellow. That's why the yellow is encapsulated inside the blue, a bit like in your game. All of this is just because of Ukraine, right? I mean, that's why we do the blue-yellow. These are all fake. These are fake colors. No, they're fluorescently dyed. It's real data. But look at this horse. So this is real data. It's doing its thing. It's a checkerboard. We change the program, and all the yellows rearranged to be in the middle, surrounded by the blue. Isn't that cool? So what's nice, this really shows you why you should only use emulsions or mayonnaise, because even if you're a compact structure like a checkerboard, you still have enough degrees of freedom to change partners and change conformation. So if you're interested in folding, you can go from one to the other. Okay, here is quantitatively, you can show that you can swap so the yellow neighbors and the blue neighbors swap around. That's what I showed you. But even more interestingly than that, the checkerboard is a floppy structure. And there are, so we've been looking at the vibrational modes of these little horses or whatever poodles that we make. And what's interesting, some of them are rigid and other ones are solid, otherwise are floppy. And we've been looking, we've been looking at the autocorrelation from what's the probability of finding a neighbor next to you, and you see that this peanut shape is kind of diffuse when it's a checkerboard, and you see that when it becomes this structure of yellows surrounded by the blue, it can make a triangular lattice instead of the square lattice, and so you see that the peaks appear, and you end up making a rigid hexagonal lattice. And by changing the DNA program from the enzymes, you can switch between floppy and rigid, floppy and rigid, floppy and rigid. So it opens another material science question, which is like, let's do rheology. Let's look at viscoelastic properties. Let's make this stuff flow. And I'll show you, this is like, whoa. I'll show you that if you zoom out, now you do another trick in the lab, and you take a solid particle, this red guy here, and you put all the enzymes anchored to it. So the only way for you to see any change in your self-assembly process is when the solid anchored enzymes produce the stuff and diffuse away. So you'll see a diffusion front of the strand that you need to stick the particles together. So we claim that we have spatio-temporal control. So once you can control space and time, you start to have Turing patterns, you start to have the probability, the possibility with your mayonnaise to start making things that evolve in both space and time in a way that you can more or less control using these DNA circuits. Okay, so we're really at the stage of proof of principle and showing that it can work. Like, I don't have something super funky that we've actually made. So I'll just show you, this is the race between the yellow strand and the blue-yellow. The solid colloid is here. One diffuses up, the other one diffuses down. Gravity is actually into the board. But what you see is that they kind of, you have mixing right in the middle, and you have segregation. And so we call this morphogenesis, but it's, of course, nothing to do with anything morphogenetic. But at least the idea that you could play with organoids and like where you tell, like the idea that you have a concentration gradient of something that instructs cells where to go, that principle, that concept is kind of there. Okay, conclusions and outlook, just to show you these conformational, the idea of the conformational change in situ, the idea that you can encapsulate something and let go of it, the idea that you could make colloidal gels with these architectures that are floppy and rigid. And I want to end this class by saying that unbeing dead is not being alive, so we're not claiming that we're making artificial life, but we are claiming that we're unbeing dead. So thank you for listening.
Questions? Emmanuel.
Sorry, technical. Yeah. Thank you very much for our lecture. Very
You're
nice.
welcome.
Really, the video is amazing. I have two questions. The first one is if your aggregation process is diffusion limited, what is the process involving the merge of the clusters when the clusters are merged and increase in time?
Mm-hmm.
Another one, did you measure, for example, the typical cluster size of the system and how this cluster increase during the time, for example?
Mm-hmm.
It's I don't know, power law behavior, something like that.
So yes, what's the rate determining step? There's a few time scales in the system. One is the diffusion time scale of the colloids themselves. One is the time at which these enzymes are producing, and there's a sort of precipitous yay or nay binding when you have a threshold amount of the DNA to trigger the binding, because even if you have some DNA, but it's homeopathic, you're not going to see adhesion because entropy will still favor being unbound. So you've got something on the order of minutes for the production of the strand. You've got another process which is governed by diffusion, which is, 1 micron squared per second for a micron-sized colloid. So we know the numbers of what the diffusion constant is, and if we look at it, we were hoping that we would see something cool like cooperative, like the more I produce, the more of something happens. But actually we don't. We see that either the switch is on, sort of instantaneous, and it's really diffusion that wins. It's like whatever we see seems to be dominated by the time it takes for the strand to diffuse to where it needs to bind. So we haven't seen something super cool that's not that. Then was your other thing? Oh, the growth rate of the clusters. That's just like how quickly. I think you're thinking about colloidal aggregates, which are the solid colloids, which are fractal, which have a fractal dimension, which grow by diffusion-limited aggregation and have this scale-free character. Here it's very different. Here it's basically you're growing little crystals because they are either square or triangular based on the matrix. But then you're just seeing the rearrangement of these crystals over time, and they scale with the area, as they recruit more.
But did you get some typical exponent of this growth?
We haven't measured it, but for instance, if we change the volume fraction of the droplets with which we start, we can end up with a percolating network of these clusters, and I haven't shown you. This is in the gel limit, and I didn't want to confuse you with that. Yeah.
Okay, very interesting. In the interest of time, we have to go to coffee break and be back by 4. But please ask questions during coffee break while Jasna is still here. So let's thank Jasna again.
Thank you so much. Thank you.